As we're collecting, we also are going to have some Twitter questions. We have a few already, but Aidan, did you want to just kick things off with anything before we collect some of the questions?
Well, we have a question already, which is an interesting question, and it's a question for Dr. Cravat. Endogenous cannabinoid release, is it affected by either acute or chronic THC administration?
Not to my knowledge, but I'll let Ken and Daniela comment on that. Are you aware of any evidence that endocannabinoid production is affected by exogenous cannabinoid uptake, which I believe was the question?
Do exogenous cannabinoids affect the release of endocannabinoids?
Not that I'm aware of.
This is a very, very important area of research because the endocannabinoid system is a very plastic system. It tends to respond, as Ken has argued, tends to respond to exogenous stimuli, particularly activation of the system itself, and is very possible that the system would be engaged by exogenous drugs and respond to that engagement with adaptations. There could be persistent adaptations. We do not have data except for a very, very few cases. For example, in exposure of adolescent animals to THC causes changes in anandamide production in the prefrontal cortex in those animals later on in life. So there seemed to be a regulation, but I think a lot more work is needed to unravel it.
I think another important aspect of that might be worthwhile discussing is while they may or may not affect endocannabinoid release, what about on the other side of the synapse? Do exogenous cannabinoids like THC affect the amount of CB1 receptor expressed?
Very strongly. So that's very, very clear. Yeah.
And in what way does it do that? What does it do to them?
Well, you'll tend to either downregulate number or downregulate coupling. So basically make them signal less efficiently.
So essentially the endocannabinoid function would be diminished because
Correct.
the receptors are diminished rather than acting on release of the agents.
Right.
Okay, how does the distribution of CB1 receptor in the brain differ in early prenatal development versus in adults? What's in the literature?
I guess that's for me. So actually, and we'll hear much more about this later in the symposium, cannabinoid receptors, CB1 receptors particularly play a very interesting role in neural development, and they undergo very interesting shifts. So in embryonic development, in focus on cortex, you have development, you have neurogenesis, you have division, you have migration, then you have process development, synapse specification. So during synapse specification of glutamatergic projection neurons, particularly, you have CB1 receptors and Maglipase very involved in the extension of those processes and formation of synapses. So normally, as all of us has said, that in adults you have postsynaptic production of endocannabinoids acting on presynaptic receptors. During development, the whole endocannabinoid system seems to be intact in the axon, involved in axon elongation and finding the appropriate target. So CB1 receptors have a very interesting role in development, and they undergo significant changes migrating from endocannabinoid production presynaptically to endocannabinoid production postsynaptically after development is maintained, as one example.
If I may add one more thought to this, in adolescence, again, there is clearly an increase in the number of cannabinoid receptors, at least in the prefrontal cortex, as well as the coupling to GI, along with an increase in anandamide. So this is quite important because there is data out there suggesting that the endocannabinoid system is crucial to key aspect of adolescent behavior, particularly risk-taking, which is, of course, cognate to drug addiction, but also things that are like social behavior, which are, of course, some of the things that we associate typically with the behavior of an adolescent. Likewise, the endocannabinoid system seems to sort of also change in old age, where other forms of plasticity occur, and the system also appears to be sort of monitoring these changes and changing in parallel with it, with the rest of the brain.
Can I ask a question just about the prenatal aspect with exogenous cannabinoids? Does it change the sort of migration of those neurons early in development? Is there any change associated with exogenous availability or with exogenous cannabinoids in the system at early development?
Yeah, I mean, there's a lot of animal data on that. It's a little bit clouded because a lot of those studies have used high-efficacy agonists, which don't really mimic the human situation, but the effects are widespread. If you go to the human epidemiological literature, there's certainly 2 studies that suggest there are enduring consequences of prenatal use, and they tend to focus on prefrontal cortex abnormalities.
Does anyone know whether in endothelial cells, the cells that line blood vessels, whether CB2 is constitutively expressed or whether it's inducibly expressed?
So in cortex, there's good RNA-seq data showing that you have CB2, at least mRNA, in cerebral endothelial cells under basal conditions. So it's there even under non-stimulated conditions, non-inflammatory.
But that's mRNA, not protein?
That's mRNA. Antibodies aren't terribly good, which many we can talk about at the coffee break.
Okay, and a question about cannabidiol. It's a question as to whether it's an inhibitor of MAGL and FAR, and could you, when discussing this, could you address whether it's an inhibitor at concentrations which are ever achieved either during marijuana ingestion or even or potentially under pharmacological conditions?
I'll take this one. Cannabidiol is the epitome of polypharmacy. It does everything, okay? Which means that it does nothing, or we are really wrong as to what it does. But it does do, among the many things that it does block anandamide transport into cells, and it does block FAR, but at high micromolar concentrations. I'm talking about 100, 150 micromolar EC50s. Now, this said, in the one clinical trial that I am aware of where these things have been tested, when CBD was given to patients, the levels, this was psychotic individuals, subjects with schizophrenia, the levels of FAR substrates were measured in those patients, and they were found to increase with the administration of cannabidiol, and also to be positively correlated with symptom relief. So if there is no direct, and those experiments suggest that there is some degree of FAR inhibition occurring because all FAR substrates in plasma of those subjects were increased by treatment with cannabidiol, and also suggested that blockade of FAR could have therapeutic utility. But as I said, CBD is the epitome of polypharmacy, so I think that we cannot draw such a simple conclusion just based on that one piece of data.
Question for Dr. Mackey. Is there any evidence that THC and/or CBD acts as an allosteric orthosteric pair at the receptor, I presume, to reverse cell death compared with THC alone? Does CBD act as an allosteric modulator at CB1?
Yeah. So hard to say. I mean, because the data I showed, that's again a recent paper from Eileen Donovan Wright's lab. The evidence is all correlative. You know, you have cell death and you have these differences in allosteric modulator and signaling, but whether one's causing the other is not determined. If that answers the question. It's basically very recent work which really needs to be explored further, I think is the safest thing to say.
The next one is a very important question, but I don't know whether it's going to be something that can be answered quickly, but here's a challenge for and it's a question of the endocannabinoid system as a homeostatic system. Can you talk about it being a regulator of regulators?
I'll start. I might as well start. And please, interject as you see fit. This is it's almost a philosophical question, but I think it's a very good question, as a matter of fact, because I personally believe that the endocannabinoid system is indeed a homeostatic system, homeostatic and allostatic. That is, under normal circumstances, in a healthy individual, the system can play an important role in controlling, if you're talking about 2AG, synaptic function, or if you're talking about an anandamide, the response to stress. But if the system becomes pathological, so for example, after prolonged exposure to THC, I think it can become also allostatic. So it's homeostasis that is no longer adaptive, is maladaptive. And that is something that we always need to keep in mind. That something that is meant to regulate homeostasis can play a role also in allostasis.
And another question for Dr. Piamelli. Do COX-1 inhibitors, these are drugs such as non-steroidal anti-inflammatory drugs, aspirin and ibuprofen, do COX-1 inhibitors have any effects on 2AG or anandamide levels?
Yeah, okay. Hopefully won't reverberate. Do COX inhibitors have any effect on 2AG and anandamide? COX-2 inhibitors do. COX-1 inhibitors don't. The COX-2 is, first of all, COX stands for cyclooxygenase, which is an enzyme that makes prostaglandins. And utilizes arachidonic acid predominantly as a precursor. However, endocannabinoids can be also used as precursors, but only by the 2 isoform of cyclooxygenase COX-2. The 1 isoform has a very narrow substrate binding domain. It doesn't allow the entry of anything else but arachidonic acid. Some mixed COX-1, COX-2 inhibitors, such as fulbiprofen, for example, ibuprofen to a certain extent at high doses, but most importantly carprofen, which is not used in people, they will block COX-2 and 5 at the same time because of this similarity. And so they will affect the levels of endocannabinoids through that pathway by blocking 5.
Right, okay. So here's a question about THC as a partial agonist. We know it's been discussed that it's a partial agonist, not a superagonist. If you take enough THC, does the high concentration make it become a superagonist, or would its effects be? Could it have an effect that's so long-lasting that it has downstream effects that would be like a superagonist?
To answer the first part of the question, no. That's kind of the definition of a partial agonist. With a given receptor density, you'll only get so much effect. But of course, that's looking at a simple dose response. When you look at the whole animal and you give a huge amount of drug and it lasts a long time, there's a lot of other things that goes on. So, you'll basically get long-lasting effects, very stoned, but it will still be qualitatively a little bit different from taking a spice compound for a very long time.
Yeah, I'm sorry, just if I may interject. I think it's really important to understand this concept because this is a crucial concept in cannabinoid pharmacology, and I'm so glad that Ken developed it so well because it's really very, very important. But a partial agonist is a partial agonist is a partial agonist. Yeah. So THC will not do what CP55940 does. That's about it. But then if you take a whole lot of THC over time, stuff can happen, but it's not the same.
If you combine the two, the superagonist with the partial, is there any additive effect here, or how does that antagonistic?
Yeah, it can actually antagonize. Yeah,
THC would actually is the superagonist
potentially.
So FAR inhibitors have at least preliminarily been shown to reduce cannabis withdrawal in one human study. It's still preliminary data, but it looks interesting. Somebody's asking whether Maglipase inhibitors might also be used to reduce cannabis withdrawal.
Yeah, so I think the preclinical work is supportive of that hypothesis. I think at least in a broader assessment of Maglipase inhibitors and their potential to mitigate drug abuse. A lot of work from Aaron Lichtman's lab showing that Maglipase inhibitors can suppress some of the withdrawal symptoms, for example, of opioid dependence. Cannabis dependence is a little bit harder to model in rodents, but it can be modeled, and I think those studies are ongoing. But I do think that's an interesting hypothesis, and relates back to what Daniele was talking about, homeostatic balance, that I think trying to re-achieve that in the context of exogenous drug abuse with this system is an intriguing area to pursue.
Yeah.
If we look at the work that Meg Haney has been developing over the years at Columbia University over this, is really fascinating, is that cannabis withdrawal is systematically accompanied by all the things that Phi inhibitors alleviate. There is irritability, there is anxiety, there is depression. All these things are clearly drivers. This negative dysphoria is clearly a driver in the continuing use of cannabis. And the fact that Phi inhibitors have such an opposite effect tallies well with the Cyril de Souza's data you were referring to before, suggesting fairly substantial clinical trial efficacy of the Pfizer compound in CUD, in cannabis use disorder.
Yeah, follow up on that briefly too. I mean, the origins of that trial could go back to Pfizer's original study on looking at sleep-wake cycles in healthy subjects, where if you take healthy subjects and poke and prod them in a sleep trial, they undergo transient insomnia, not surprisingly, and you can look at the effect of drugs on rectifying that transient insomnia, and that's where the first effects on mitigating these sorts of perturbations in basal sleep-wake cycle was observed. And I think that, so it's in a way, it was somewhat of a replication of that result in the context of marijuana dependence, right, where sleep withdrawal is a major issue. So
we have a question coming through by Twitter. It's a question about CBD affecting THC at CB1. I think it's probably important to consider the concentrations. In the concentrations that people who consume marijuana would be exposed to THC and CBD in its different forms, does CBD ever reduce the psychoactive effects of THC by preventing its interaction at CB1?
Okay. There's clear evidence going back many years that the ratio of CBD to THC affects the psychoactivity of cannabis. I mean, that's very clear data. So yes, how it does, it's not clear. But I think the CBD effects are more interesting when considering CBD as a therapeutic agent, say in Dravet's or something, because normally CBD levels are not very high. Okay. So as most of you know, the breeding of cannabis over the last few years has decreased the amount of CBD, well, increased the amount of THC. But even if you take THC, CBD at relatively equal levels, CBD levels are substantially lower, and Marilyn can address this more. But if you take CBD therapeutically, you can push the levels pretty high in terms of the amounts that are used for Dravet's, because you don't have the psychoactivity. So there, the concentrations in the graph that I showed, which are 50 nanomolar or so, 100 nanomolar, are easily achievable in serum.
So under conditions of marijuana smoking?
So conditions of marijuana smoking, clearly you have effects, whether they're mediated through negative allosteric modulation or serotonin receptors or the 5 dozen other targets of CBD at those concentrations. One can't say.
And the effects
are generally to decrease the strength of the high or more of the negative consequences of the high to drastically oversimplify the data.
Okay, 2 more questions. So, Dr. Piamelli, do you think that an endocannabinoid deficiency could potentially cause ailments in its own right?
Yes.
And often one sees a deficiency, a natural disease which can be associated with a deficient enzyme of some kind. Do we know of any natural diseases that are caused by deficiencies in any of the endocannabinoid synthetic enzymes?
Well, to my knowledge, there are no human knockouts, if you will, of either the major endocannabinoid metabolizing enzymes, biosynthetic or degradative. But there is a common polymorphism in FAW that has been linked to the potential for drug abuse, as well as to a variety of anxiety-related syndromes. And so I think those studies are now becoming more frequent now that the polymorphism has been identified and prospectively analyzing them in rigorous clinical studies. But certainly that is not a complete disruption of the enzyme, it's a partial disruption, and our understanding of its impact on FAW and endocannabinoid signaling is in early days, but there is at least some human genetic opportunity there to study that arm with that polymorphism. So.
And finally, is there any evidence that agents that are pharmacologically active at CB2 exclusively should be scheduled in any way? Do they have any psychoactive effects that would make them abusable?
Not that I know of. Not quite a short answer as Daniele, but pretty much.
Great. Well, please join me in thanking our first panel.